Gregor Mendel, born Johann Mendel in 1822, emerged from humble beginnings in a small village in Silesia Tabide. His family were farmers, instilling in him an early appreciation for the natural world and its intricate workings. The young Johann displayed exceptional intellectual curiosity from a very tender age. He often spent hours observing the plants and animals around his family's modest homestead. His parents recognized his keen intelligence and made significant sacrifices to ensure his education. Despite financial hardships, he pursued his studies with remarkable diligence and dedication. He attended the Piarist Gymnasium in Lipnik nad Bikvu, where he excelled in his academic pursuits. Later, he enrolled at the Philosophical Institute in Olomouc, further broadening his intellectual horizons. The financial strain of his education, however, became increasingly burdensome for his family. This challenging situation led him to consider a path that would allow him to continue his intellectual development without financial worry. In 1843, at the age of 21, he made a pivotal decision. He entered the Augustinian Saint Thomas's Abbey in Brno, which was then part of the Austrian Empire. Upon entering the monastery, he adopted the name Gregor, a name that would forever be associated with groundbreaking scientific discovery. The abbey was not merely a religious institution. It was a vibrant center of intellectual life and scientific inquiry. Many of its monks were actively involved in teaching and research, fostering an environment conducive to scholarly pursuits. This intellectual atmosphere provided Mendel with an unparalleled opportunity to pursue his scientific interests. He was particularly drawn to the study of heredity, a field that remained largely mysterious at the time. The prevailing theories of inheritance were vague and often contradictory. Many believed in blending inheritance, where offspring would simply be an intermediate mix of their parents' traits. Mendel, however, suspected a more precise and predictable mechanism was at play. His superiors at the abbey recognized his intellectual gifts and supported his academic endeavors. From 1851 to 1853, he was sent to the University of Vienna to study. There, he delved into various scientific disciplines, including physics, botany, and zoology. His studies under renowned professors like Franz Unger, a botanist, and Christian Doppler, a physicist, profoundly influenced his scientific methodology. Unger's work on plant hybridization and Doppler's emphasis on empirical observation and mathematical analysis shaped Mendel's approach. He returned to the abbey in 1853, equipped with a rigorous scientific training and a burning desire to unravel the mysteries of heredity. He began teaching natural science at the local technical school, a role he fulfilled with great enthusiasm. However, his true passion lay in experimental research. He meticulously planned a series of experiments that would revolutionize the understanding of inheritance. He chose the common garden pea, Pisum sativum, as his model organism. This choice was not arbitrary. Peas possessed several advantageous characteristics for genetic study. They were easy to cultivate and had a relatively short generation time. Crucially, they exhibited several distinct, contrasting traits, such as tall or dwarf height, round or wrinkled seeds, and green or yellow pods. Furthermore, peas could be self-pollinated or cross-pollinated, allowing for controlled breeding experiments. Mendel embarked on his groundbreaking experiments in 1856, dedicating years to careful observation and meticulous record-keeping. He started by establishing pure breeding lines for each of the 7 traits he intended to study. This involved self-pollinating plants for several generations to ensure that they consistently produced offspring with the same trait. For example, he ensured that tall plants always produced tall offspring and dwarf plants always produced dwarf offspring. Once he had his pure breeding lines, he began his hybridization experiments. He cross-pollinated plants with contrasting traits, such as a pure-breeding tall plant with a pure-breeding dwarf plant. These initial crosses produced the first filial generation, or F1 generation. To his surprise, all the F1 offspring exhibited only one of the parental traits. For instance, when he crossed tall and dwarf plants, all the F1 plants were tall. The dwarf trait seemed to have vanished entirely. This observation directly contradicted the blending inheritance theory. Mendel then allowed the F1 generation plants to self-pollinate. The offspring of this self-pollination constituted the second filial generation, or F2 generation. In the F2 generation, the seemingly lost trait reappeared. He observed a consistent ratio of approximately 3:1 for the dominant trait to the recessive trait. For example, in the F2 generation of the tall and dwarf cross, about 3/4 of the plants were tall, and 1/4 were dwarf. This precise numerical ratio was a critical piece of evidence. Mendel repeated these experiments for all 7 contrasting traits, and each time, he observed similar patterns and ratios. He meticulously counted thousands of individual pea plants and their characteristics. His rigorous quantitative approach was unprecedented in the study of heredity. Based on his extensive data, Mendel formulated several fundamental principles of inheritance. He proposed the existence of heritable factors, which we now call genes, that determine specific traits. He suggested that each parent contributes one such factor to their offspring. His first law, the law of segregation, states that during gamete formation, the 2 alleles for a heritable character separate, or segregate, from each other, so that each gamete carries only one allele. His second law, the law of independent assortment, states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. This means that the inheritance of one trait does not influence the inheritance of another. For example, the inheritance of seed color is independent of the inheritance of seed shape. These laws provided a clear, mechanistic explanation for how traits are passed from one generation to the next. Mendel presented his findings to the Brunn Natural History Society in 1865. His lectures, delivered at over 2 separate meetings, detailed his experimental design, results, and conclusions. The audience, composed primarily of local naturalists and scientists, did not fully grasp the profound implications of his work. His quantitative approach and the abstract nature of his factors were perhaps too far ahead of their time. The scientific community of the mid-19th century was not yet ready for such a precise, mathematical understanding of biology. In 1866, he published his seminal paper, Versuche über Pflanzenhybriden, Experiments on Plant Hybridization, in the Proceedings of the Natural History Society of Brunn. This paper laid the foundation for modern genetics. Despite its publication in a respected scientific journal, the paper received little attention from the wider scientific community. It was cited only a handful of times in the decades following its publication. Mendel continued his hybridization experiments with other plants, including hawkweed, but these proved more complex and did not yield the clear-cut results he had observed with peas. The complexities of hawkweed genetics, involving apomixis, asexual reproduction through seeds, obscured the patterns he sought. In 1868, Mendel was elected abbot of the monastery, a position that brought with it significant administrative responsibilities. These duties increasingly diverted his time and energy away from his scientific research. He became deeply involved in managing the abbey's affairs and engaging in local politics, particularly concerning taxation issues. His scientific work gradually receded into the background. He never received widespread recognition for his groundbreaking discoveries during his lifetime. passed away in 1884 at the age of 61, without knowing the immense impact his work would eventually have. His contributions remained largely unnoticed for over 3 decades. The scientific world was simply not prepared to appreciate the significance of his meticulous experiments and revolutionary insights. The rediscovery of Mendel's laws occurred independently by 3 different European scientists in 1900. Hugo de Vries, Karl Correns, and Erich von Tschermak, working on their own hybridization experiments, independently arrived at similar conclusions. As they searched the existing literature, they each stumbled upon Mendel's 1866 paper. They were astonished to find that Mendel had already articulated the very principles they were discovering. This simultaneous rediscovery marked the true beginning of the science of genetics. The year 1900 is often considered the birth year of modern genetics, thanks to this pivotal rediscovery. Mendel's heritable factors were soon linked to chromosomes and the concept of genes. His work provided the missing link in Darwin's theory of evolution, explaining the mechanism of variation and inheritance. Without Mendel's foundational work, the rapid advancements in genetics and molecular biology in the 20th century would have been impossible. His legacy continues to influence biological research to this day. The principles he uncovered are taught in every biology classroom around the world. His story is a testament to the power of careful observation, rigorous experimentation, and quantitative analysis in science. It also highlights how revolutionary ideas can sometimes take time to be fully appreciated by the broader scientific community. Mendel's pea experiments remain a classic example of scientific inquiry. His meticulous approach set a new standard for biological research. He demonstrated that complex biological phenomena could be understood through simple, well-designed experiments. His work laid the groundwork for understanding genetic diseases. It also informed agricultural practices, leading to improved crop varieties. The Augustinian monastery in Brno, where he conducted his experiments, is now a place of pilgrimage for scientists. His small garden plot, where the pea plants once grew, is preserved as a historical site. The scientific community eventually recognized him as the father of genetics. His name is synonymous with the fundamental laws of heredity. The impact of his work is immeasurable. He truly changed our understanding of life itself. His quiet dedication in the monastery garden yielded insights that continue to resonate. The simplicity of his experimental setup belied the profound nature of his discoveries. He meticulously documented every single cross and every single offspring. This level of detail was crucial for identifying the precise ratios. His patience was extraordinary, spanning many years of repetitive work. He faced skepticism and indifference during his lifetime. Yet, he persevered with his scientific quest. His legacy is a powerful reminder of the importance of basic research. The world eventually caught up to his genius. His work is a cornerstone of modern biology. Every time we discuss dominant and recessive traits, we are echoing Mendel. Every time we analyze a Punnett square, we are applying his principles. His contributions are foundational to biotechnology. They are essential for understanding human health. They are vital for agricultural innovation. The story of Gregor Mendel is one of quiet brilliance. It is a story of a monk who, through humble pea plants, unlocked the secrets of inheritance. His experiments were elegant in their simplicity. His conclusions were revolutionary in their implications. He truly founded the science of genetics. His work continues to inspire new generations of scientists. The pea plants in his garden became symbols of scientific breakthrough. His careful crosses revealed the discrete nature of hereditary units. He showed that traits are not blended but passed on as distinct entities. This was a radical departure from contemporary thought. His quantitative data provided irrefutable evidence. He transformed biology from a descriptive science into a predictive one. His insights paved the way for DNA discovery. The double helix owes a debt to Mendel's P's. His work is a testament to the scientific method. He is a giant in the history of science. His name will forever be etched in the annals of biology. The profound impact of his work cannot be overstated. His dedication to scientific truth, even in isolation, serves as an enduring inspiration. He exemplified the spirit of scientific inquiry. His careful methodology is still a model for experimental design. The clarity of his thinking allowed him to see patterns where others saw only complexity. He truly was a visionary ahead of his time. His work laid the foundation for all subsequent genetic research. From understanding human diseases to improving crop yields, Mendel's principles are indispensable. The quiet monk in his garden changed the world. His legacy is a beacon for scientific discovery. He proved that even the smallest organisms can reveal the biggest secrets. His experiments were a triumph of intellect and perseverance. The world owes a great debt to Gregor Mendel. His name will forever be synonymous with the origins of genetics.